Okay, the reason why I'm here is that I work with a person called Jay Fireman, who published a book called The Biology of Religious Behavior. We have been working on that together. I wrote a chapter in his book, and recently he sent me an email suggesting that I might present my work here. So I sent in my abstract, and I just trust the organizers that I'm not completely off. So, what is this all about? I have been engaged in a very long search for patterns in behavior, really since more or less 40 years. And it was carried out first at the University of Copenhagen, then at the Museum of Natural History in Paris, then the University of Paris 5, 8, and 13, psychology and biology, and then at the University of Iceland, where I come from. There's been a close collaboration with research teams within ethology, psychology, psychiatry, and neuroscience at a number of other universities in USA and Europe, especially the University of Chicago, with whom I have worked for over 30 years. They use these methods since then for analyzing human interactions. We also have a so-called MASI university network around this model that have been developing. There are about 24 universities in that now. And more recently, and I think maybe most interestingly for me, there's been close collaboration recently with the Barbraham Institute and the zoology department of the University of Cambridge, where from we have neuronal data. From the beginning, in the mid-70s, the focus has been on formulating, guessing, and discovering, searching for some general structural aspects of behavioral dynamics. For this, real-time structural concepts, pattern types, detection methods, and algorithms have been developed and implemented in special-purpose software called Theme that I have written over the last 35 years or so. Okay, this does not reflect the historical chain of events, but it's as I begin to see things in the last few years, trying to get an overview of the situation. So you see, according to this leading astrophysicist, Martin Rees, there are basically 6 numbers that determine the structure of the universe. Some of these forces pull things together, others keep them apart. So, thus, in the history of the universe, countless levels of hierarchical patterns of patterns are formed, not simply by clumping, but through structured clustering. So, cosmic evolution and structured clustering, hierarchical self-similar patterns, pseudofractals. Pseudofractals will come back to this. It's just give a quick background of why I think the principles that are being used here are really all over the place. So we come from this tiny irregularities, asymmetries, or structured hierarchical clusters of atoms and stars and galaxies, from pure energy to ever more complex atoms and molecules such as DNA, proteins, the cornerstones of life on Earth. And here we have a simulation snapshots of the evolution of the universe from that point of view. Fractals, the patterns that I've been proposing seem to be in the family of statistical fractals or pseudofractals. Whether they are or not is not crucial, but it really seems so. And here is a statement about such phenomena. Physical fractals typically display statistical self-similarity over scales differing by just a few factors of 10. They are not endless as in mathematics, obviously. Nonetheless, as Mandelbrot first observed, it's extremely useful to recognize the fractal properties of natural shapes. In this context, both for the universe and for these things, this book is of particular interest, and it's recently been shown that the universe, almost as far as we know it, has a fractal dimension. It's like a fractal object. It has been published in Nature. Okay, here's another astrophysicist who thinks that you are not dealing with simple elements that cluster, but we are dealing with fragments of many different kinds. So we get many different kinds of clusters And this gets very close to where I'm going. Fractal dust grains are created from a coagulation of smaller subunits, starting with the monomers. Two monomers collide to stick to make a dimer. The figure above shows a process where two dimers collide to make a tetramer, two tetramers collide to make an octamer, and so on. This produces what I call, he says, CL fractals. Now to behavior, where we have the temporal dimension. So why search for repeated behavior patterns? If we cite Francis Crick, one of the discoverers of DNA, he says, another key feature of biology is the existence of many identical examples of complex structures. And the person who is called often the father of human ethology, I. Beibelsfeld, wrote this as the first words of his book, Ethology, the Biology of Behavior, in 1970. Behavior consists of patterns in time. Investigations of behavior deal with sequences that, in contrast to bodily characteristics, are not always visible. And here comes the trick, the problem. We have lots of behavior in front of our eyes. We have some sense that there is a lot of structure, but we just cannot put our finger on it. We cannot see it. So we're in a bit of a similar situation when people looking at the stars. There may be structures out there. We may sense some of it, but we need artificial means of detecting it really. And without that, we would not know much about anything, actually. So how do we go about that for behavior? And if you take a look, most of, for example, in psychology, almost all of psychology is about questionnaires. Nobody is observing behavior. People are analyzing questionnaire data. But still, it's totally traditional viewpoint in all of behavioral science that behavior consists of repeated hierarchical syntactically constrained patterns. Whether you take linguistics, ethology, behaviorism, anthropology, social psychology, cognitive science, you have scripts and plans and routines, etc. So what seems to be needed, or has been needed for a long time, is more recognition of the hiddenness of many such patterns and new definitions and corresponding detection algorithms and tools, software, for the discovery. Here's just a tiny example to show how terribly bad we are at detecting such translation symmetry, as it's also called. The two axes are exactly the same. It helps to put some color. It helps to put some binary tree on it, but we don't really see it very clearly unless everything else is removed. And if we go back, we practically have the same problem again. And this is, of course, ridiculously simple data. Now you should think that you could just go to the next statistical center, as I had beside me in the beginning and still have easy access to, and just ask for a program that will pick this out for you. And strangely enough, it still doesn't exist, and I just don't get it. Okay? So, what is the input to the algorithm that detects this kind of patterns? The input to the detection algorithm is just a set of series, labeled series. It's a very simple mathematical object. They are all within this interval. I have to be terribly untechnical and fast because we have to say a lot in practically no time. So this is the pattern definition. With the initial series, the labels as simple as T patterns within 1 to T. A T pattern is an ordered set of T patterns. It's recursive, where you have the X's stand for the different T patterns, and the approximate DT, 1, 2, etc., stand for the approximate, highly constrained temporal distances between them. It recurs when it recurs. It recurs such that each of the time distances dt varies significantly less than expected, assuming a zero hypothesis of constant probability per time unit for each xi given by this. If you just take then the variation ranges, you can represent this as a series of critical intervals. And that's how the the detection proceeds. It's possible to take any such sequence and split it into two and make the requirement that there is a critical interval relationship between the two halves. You can split that down to the terminals, but the detection works the other way, from the bottom up, and builds the trees. But to avoid combinatorial explosion, there is an evolution part of this algorithm that takes care of that. This is all I can say about it here. It's of course far too short. This is real data. 2 children playing together, 82 event types, approximately 40 per child. It's 13.5 minutes. So here are these lines of dots. That's all the algorithm gets. And it's supposed to find out if there is any pattern between the children that recurs. And what it finds was a total shock when I found it a long time ago. It showed that actually throughout 100% of the observation time, they were performing 4 times the same pattern with very little variation. You can see this includes 25 of the 82 series. Usually when you analyze such data with traditional methods, you're lucky if you find sequences of 3 and 4, something like that. And here you can see this fractal look of it. There's 4 translations. Okay, then people would say in the beginning, well, if you had randomized all your data and you'd done the same, you might have found something similar. So since 1981, there has been this module in the software that does that. It shuffles all the data and runs the same algorithm again. And recently, in relation to pattern detection of this kind in neuronal data, I added something more than the shuffling. Each of these series that goes in is not shuffled, but is rotated, shifted a random amount, so that each series maintains its structure, but the relationship between the series has been randomized. And here you can see the enormous difference between what you find in the real data and what you find in the randomized data. 5, okay. And here the number of standard deviations for this kind of data often goes up to hundreds of standard deviations and even thousands. The system has been extended with many different concepts. I have no time to get into that of course. I'll just show you, this is my favorite concept, which is I call the T-packet. It is a T-pattern with repulsion and attraction zones, and defined by associates that tend to be there or tend not to be there. Here is neuronal data. Same thing, neuronal interpret data registered from a chip in a rat olfactory bulb. Occurrent spike series of 83 neurons and 2 breathing in-out series. They are up here. There has been enormous difficulty in discovering any patterns in such data, and there's a new article about it now, where there's much. Some people just think of what's happening is just a flow. Others think there are patterns, and the leading person in that has made a very simple algorithm where he has very big problems trying to show that what he gets out is different from random. I will show you how different it is from random when you do it this way. And there is also external validation on top of the statistical validation, in that breathing comes into lots of these patterns. Further external validation. This animal here, this is average difference between random and real. And you see that difference is gradually fading, both here and here. But this animal here is breaking down, and here it has to be stopped because it's breaking down completely. So when it's breaking down, it loses the difference between random and real. The patterns of interaction between its neurons seem to be fading away. Here is, if we look at the genes, we see something that almost looks like a description of T patterns. It has these patterns and intervals between them where anything can happen. Just as within a dinner, you have certain steps, but what happens in between them is undefined and can be whatever. But the temporal structure of a dinner is approximately rigid. So we have again this type of structure. So I'm practically finished. Practically, in a way, tongue-in-cheek, but still, it's in a publication. I talk about a dinner gene, and I think we just saw it used here. This could be a drawing of a gene or of a dinner. Each of the black parts are the different parts of a dinner. You see, sitting down, taking an entree, taking a main course, taking a dessert, smoking, standing up. But it can also just be a gene. And what happens in between those can be any nonsense, nothing, or a lot. And just as molecules, they are flexible to a certain extent and squeezable to a certain extent. In the same way, a dinner can be stretched, but never over a week. It has very strict limits on how much you can stretch it in time and how much you can squeeze it in time. But what struck me was that it's also functionally a gene. How did this conference begin? It began with a dinner to facilitate certain types of social processes. So when you need that kind of facilitation, you start expressing the social gene called a dinner. It has the mathematical structural properties of all kinds of human behavior, rituals or words or whatever you like, and of genes. But these are spatial. But the expression of them has an analogous function within human societies as the expression of genes has in the society of proteins, in cell city. So this is the last slide. And I couldn't help taking it, adding it. It comes from one of my talks on religion, the biology of religion. And I noticed on Open University site, where they have Nobel Prize winners who study cell city, there was an analogy between the different parts of cell city and human city. And what they did, what they called the ribosomes factories, which is strange, because most of what ribosomes create are the citizens of cell city, are the specialists of cell city. We would call that schools here in our societies, for best of my knowledge, and nobody has criticized this analogy. But that is quite a twist from factory to school. And recently somebody is criticizing the fact that our schools have become factories. And that's another story. And my father, who was a teacher, he used to say sometimes when he was not in his best mood, I work in a factory that produces, specialists. And I think I will just, you see, we have a subset of the textome of our cities, like they have a subset of the genome or the DNAs of their cities. They use a fraction of that to produce, with active shapers, different types of specialists. And we could go on for there, but that's it. Thank you very much.
Thank you. Questions?
Like a very basic question. So a T-pattern is kind of almost self-invariant pattern that is like fractal or something like that?
It is, and I think it relates to the discussions we had just before about these network analysis. I'm so old that I was one of those who was doing these first sociograms in the early 70s, and I thought that was very interesting. And I also had acquaintance with Tinbergen's flowchart descriptions of stickleback behavior. But the problem was always this binary aspect. You didn't have multi-ordinal relationships. You have A going to B, and then you have something about from B going to C. But then all information about where you came from when you came from B was lost. And that is run totally counter to what we see about the syntactic hierarchical structure of behavioral patterns, and actually of patterns all over the place, whether it's in chemistry or in the universe, as we just saw. It seems to be so extremely natural for us that things should be hierarchically organized and repeated. And things cannot combine before they exist. You cannot combine a radio and a car before you have a radio and a car. It was like with the polymers there in the beginning. So what I had to deal with was to get beyond this binary kind of networking, because it's absolutely hopeless for describing behavior in time, and which is maybe one of the reasons also. And it seems the neurons are doing the same thing. They do little patterns, where they do on many places, but sometimes they combine them, make bigger ones. But when you have these bigger ones, they may connect with other bigger ones that they do with other different combinations. And only if you do that, you can find patterns that are extremely significant, way beyond anything that we usually ask for in statistics. And now we just discovered this, that when you see